Welcome to this presentation on BioCule's DNA quality control platform. BioCule is currently looking for commercialization partners or acquirers to help fully commercialize this game-changing sample QC technology platform. Let's start with an overview of a typical DNA-based analysis workflow. We're using sequencing as the example, but the BioCule platform can be applied equally as well to other technologies like PCR or microarrays. Genetics and genomics analysis is a large and very fast-growing market. To capture this growth, companies have invested hundreds of millions of dollars into new technologies and products covering various aspects of this workflow, from preparing the sample, sequencing the DNA, analyzing the results with various bioinformatics algorithms, and, especially for clinical samples, interpreting and reporting the results. However, the crucial step of assessing sample quality is often overlooked or ignored completely. Skipping this step threatens the utility of the rest of the workflow. Poor quality samples will lead to poor, or worse, misleading results. This critical starting point hasn't seen much innovation for many years, until now. Poor sample quality is usually due to damaged DNA, which can be caused by a variety of reasons, including storage conditions, improper processing, and contamination. Some typical types of DNA damage are fragmentation, single-stranded DNA, DNA nicks, and crosslinks. Most of these types of damage are completely invisible to the leading QC platforms, making it impossible for researchers to make informed decisions about their samples. While we won't be going into all the details of the many different ways low-quality DNA samples can cause problems, I'll give a couple of examples. The most common problem is fragmentation. Sample amplification is a typical step in most DNA processing applications. However, when DNA is highly fragmented, the amplification step usually fails. Similar problems would be seen with DNA that is nicked. These failure modes would either lead to no results or, worse, incorrect results that would be open to misinterpretation. Another example of a big problem, and therefore a market opportunity, is cross-link DNA damage, especially in formalin-fixed paraffin-embedded, or FFPE, samples. As we said before, DNA amplification is a key step in many genomic applications. But when DNA is bound up with crosslinks, the DNA amplification step isn't successful. Therefore, the amplification will result in only a portion of the genome being correctly amplified. And as there is no way to know ahead of time which regions of the genome suffer from crosslink damage, the end results are very difficult to interpret. This is especially critical in the clinical market, as FFPE samples are often from patient tissue biopsies or other human tissues. There are a number of QC solutions on the market, but none of them are able to offer a complete picture of sample QC. This leads to the core problem that researchers in clinical labs are not able to know the sample quality before they run their DNA analysis, and often not even after they have completed it, leading to incorrect or confusing results. NanoDrop is one of the most widely used QC instruments. However, it's actually more of a quantity control rather than a quality control instrument. People use it to find out how much DNA they have in a sample, but it doesn't tell them anything about the quality of the sample, nothing about the level of fragmentation, amount of single-stranded versus double-stranded DNA, or the level of cross-linking. This often leads to an artificially high estimate of the amount of functional DNA in a sample. Many labs also use a bioanalyzer after the NanoDrop to QC the sample. However, the bioanalyzer only offers a one-dimensional view of the sample, and therefore can only report on the level of fragmentation. But fragmentation is only one way that DNA can be damaged. Nothing is reported on the amount of single-stranded versus double-stranded DNA, the level of cross-linking, or any other type of damage, such as DNA nicks. Despite being the gold standard for sample QC, the bioanalyzer has a lot of shortcomings and is ripe for improvement and replacement. These suboptimal sample QC platforms often result in poor, incorrect, or misleading results. This can lead to a number of undesirable outcomes, including wasted time and wasted resources. In some cases, it may even be possible to rescue highly damaged samples if the precise nature of the damage is understood. This is a highly valuable proposition to increase the value of the millions of samples stored in biobanks around the world. The solution is BioCule's two-dimensional gel analysis platform. This QC test is able to report on the level of DNA fragmentation, percent of double-stranded DNA, percent of single-stranded DNA, percent of cross-linked DNA, as well as the percent of other types of DNA damage. To show how this works with real samples, we've looked at a number of head-to-head comparisons between the bioanalyzer, NanoDrop, and BioCule across a variety of sample types. The example shown here is with FFPE samples due to their prevalence with hundreds of millions of samples stored around the world, importance as clinical samples, and difficulty in working with them due to their highly variable levels of DNA damage. The nanodrop platform is able to give us a basic level of info of the three samples, namely that there appears to be enough sample to work with. However, it isn't able to say anything about the quality of the samples. For all three samples, the nanodrop gives a passing grade. To get a better look at the quality, we switched to the bioanalyzer platform, the QC workhorse and industry gold standard. As expected for FFPE samples, we can see that all three samples are fragmented. But as the level of fragmentation isn't too severe, all three get a passing grade from the bioanalyzer. Note that for all three, the degradation patterns look indistinguishable. It's only when looking at the samples with BioCule QC platform that we start to see differences. The first sample has only 6% double-stranded DNA, with the remainder of the sample being single-stranded, cross-linked, or suffering from some other type of damage. This sample gets a failing quality grade from the BioCule platform, with the recommendation not to use it in subsequent analyses. The second sample is a little bit better, with 17% of the DNA being double-stranded. While not great, this sample gets a borderline quality grade from the BioCule platform. It would be at the user's discretion as to whether or not to continue with the sample. It would likely be replaced with an alternate sample if available. If, however, it were rare or especially important, the researcher might decide to continue using it. The third sample looks substantially better, with over 40% of the DNA in the double-stranded state. For an FFPE sample, this level of double-stranded DNA and fragmentation would get a passing quality grade from the BioCule platform. Here we have a summary of how the three QC platforms assess the quality of the samples. We then proceeded to use these samples in a real-world test. They were used to generate sequencing libraries using the Illumina TruSeq cancer panel, which was then sequenced on a single MySeq flow cell. Based on the number of aligned reads generated with these three libraries, we can see that the first sample is a complete failure, with no aligned reads being generated. The second sample, while not a complete failure, was only able to generate 42,000 reads. Of the three, only the third sample produced satisfactory results, with over 700,000 aligned reads from the cancer panel. The nanodrop platform gave incorrect calls to sample 1 and 2, only getting the third one correct, giving it a failing grade. The bioanalyzer platform also gave incorrect calls for samples 1 and 2, only getting the third one correct. Again, also getting a failing grade. Only BioCule was able to correctly assess the quality of all three samples, giving it the only passing grade for all three platforms. Now that you've seen how well the BioCule platform works, we want to show you a bit about how the technology works. BioCule has developed a proprietary two-dimensional gel electrophoresis technology. By separating the molecules across 2 dimensions instead of just 1, we're able to tell a lot more about the quality of the molecules. Specifically, we can estimate levels of double-stranded DNA, single-stranded DNA, DNA nicks, and cross-linked DNA. In the following cartoon images of the BioCule gel cartridge, we'll be using false color images with a DNA sample in green and a standard ladder in red. In this acrylamide gel, the first dimension is run under non-denaturing conditions to separate the molecules out by size, conformation, and strandedness. Prior to the second dimension, the samples are heat denatured so that all molecules become single-stranded. The second dimension is then conducted under denaturing conditions, such that the molecules are separated strictly by size. In this example, we're looking at undamaged genomic DNA. Since the DNA has a high molecular weight, it really doesn't migrate into the gel, so we end up with this large green block in the upper left corner. If we take the same DNA and digest it with a restriction endonuclease, the DNA is now able to migrate into the gel. This is what digested or artificially fragmented, but otherwise undamaged double-stranded DNA would look like. The sample migrates along the path of the DNA ladder. Now let's take a look at a damaged sample. Looking at an undigested sample, we can see 2 types of damage. DNA that is fragmented with double-stranded breaks migrates along the path of the DNA ladder, looking a bit like intact DNA that has been artificially digested. We can also see DNA with single-stranded breaks as a horizontal line. To get a better view of what's going on, we can run a digested sample in parallel. DNA with double-stranded breaks still runs along this path with the ladder, but we can also see a variety of other types of damage. Running to the left of the ladder, we see interstrand crosslinks and alpha helix DNA. When working with cDNA samples, this is also where we would see DNA-RNA hybrids. Running to the right of the ladder, we can see a diagonal line of single-stranded DNA, as well as additional types of damage, such as bent DNA, single-stranded breaks, and intrastrand crosslinks. With the use of standard imaging analysis software, this output can be summarized into a report detailing out the various types and levels of sample damage. Here's a sneak peek at what the BioCule technology really looks like. On the left is an image of one of the service units being used to run customer samples in-house. In the middle are actual gel images of samples suffering from various levels and types of DNA damage. Based on initial customer interviews, we have identified a strong interest and demand for the BioCule QC technology in a number of large, key markets. The most obvious is that of the broad life science research market, where anyone that uses DNA-based analysis has a vested interest in better QC technology, allowing them to avoid problems with low-quality samples, such as wasted time and resources. A subset of this is next-generation sequencing, one of the fastest-growing markets in our industry, which has been estimated to comprise around 2 million samples in 2015 and projected to double in the next couple of years. As the cost of sequencing has continued to fall, more and more researchers are looking to sequence old samples stored away in freezers and FFPE blocks. A better QC platform than is commercially available today would be crucial in unlocking the power of these currently unavailable samples. Another example is the clinical market, which is already fairly large and poised to grow larger over the next several years. As mentioned previously, one important subsegment of the clinical market is that of FFPE samples, with hundreds of millions of samples stored in clinical biobanks around the world. We hope this presentation has provided you with an initial overview of BioCule's state-of-the-art DNA quality control program. We would be happy to arrange a conference call or an email exchange in order to answer any questions you may have and discuss the possibilities of acquiring the BioCule technology for your product portfolio. If interested, please contact us at info@biocule.com.